Today I wanted to deliver my lecture on jaundice. Is it a disease? Yes or no? Jaundice, is it a disease? No, it's not a disease. So what is it then? Sign of some diseases. Manifestations of some diseases. Jaundice is also termed as, yesterday when there was one lecture, he told, what is this? He gave you the term. What is the term? Icterus. Jaundice or icterus. So you will not forget. While you are taking a history of a patient, you will be examining a patient. You will be studying, examining whether he or she is having icterus, means jaundice. So what you will do? You will first see the sclera, see the palm, coloration of the skin. Then how can you define jaundice? What is jaundice or icterus? It is yellowish coloration of skin, sclera, palm skin. So yellowish coloration. Sometimes it may be green. When the condition is worsened, level of bilirubin is high. Level of conjugated bilirubin is high, very high. And at that time, another pigment may come. May come means their level may rise. That is biliverdin. Biliverdin. And it is green in color. So because of presence of biliverdin in high amount, it may give, instead of yellowish coloration, there may be greenish coloration. So before we enter into the jaundice, what are the different types of jaundice, let's see. This bilirubin, biliverdin, conjugated bilirubin, unconjugated or free bilirubin, where from it comes? They come. What is their origin? The origin is, of course, hemoglobin. Of course, hemoglobin. And hemoglobin stays in, where you will get hemoglobin? In RBC. So RBC has got a life. How many days? 140 days. So after 140 days, under normal conditions, suppose there is no disease, what will happen to this RBC? One is elderly RBC. It is becoming non-functional. Its membrane is usually flexible, no? So that it passes through the capillaries. So, within the cell membrane, there are some cytoskeleton, you know that? So, this cytoskeleton actually make changes in the shape of the RBC and help in transportation of passage of RBC through the capillaries. For those who are new, they should know the diameter of the capillaries is lesser than the size of the RBC. So how it can pass? It is passing through the capillaries. How it's passing through the capillaries? By changing its shape. By changing its shape, and it is possible because of flexibility of the membrane, and this flexibility is rendered by cytoskeleton present in the red cells. Okay, so this cell membrane now become rigid. So they will not be able to pass through the capillaries. So immediately this type of RBCs will be trapped by bone marrow, because blood will also be passing through this. So it will take up the old RBCs, or my liver can also take up, or my spleen, it can also take up this old, damaged, or going to be damaged non-functional RBCs by these organs. What are these organs? Liver, bone marrow, spleen, and lymph glands. So there are so many lymph glands throughout the body. Therefore, preventive function in our body. So they can trap these old senile RBCs. Then what it will do? There are macrophages. What? Macrophages. They have the ability of phagocytosis. This macrophage comes under reticuloendothelial system. We'll learn it later. Well, we'll be learning about immunity of our body, then you will find what are the different kinds of macrophages, different kinds of reticuloendothelial cells. So one type is macrophage. In the liver cell, there are Kupffer cells. These are the macrophages. So what they do? The macrophage present in the bone marrow, or in liver, or in spleen, or in lymph glands, what will they do? They will capture this senile or old RBCs and break down the cell. After phagocytosis, taking in the cell within their cytoplasm, they will break it. What will come out? Hemoglobin will come out. Hemoglobin will come out. So we'll get hemoglobin. We'll get hemoglobin. Now what will happen to this hemoglobin? Hemoglobin is made up of heme and globin. So it will be splitted. It will be splitted. So what will be formed? Heme will be formed and globin will be formed. Now we know globin is a protein, and proteins are made up of amino acids, and they contain so many essential amino acids. The new blue shirt, sky blue shirt, yeah. I'm asking you one question, simple question. What is essential amino acid? What are essential amino acids? Okay, don't know. So listen carefully. Proteins are made up of amino acids, and they are linked together by peptide bonds. Okay? If by the peptidase enzyme, if these peptide bonds are broken, then amino acids will be liberated. And this protein, there are so many amino acids. There are so many amino acids. And out of this amino acids, many can be synthesized in my body, but some are not. Some are not. But they are essential for my body. Essential component of so many, of almost all the proteins, different kinds of protein we are having. So all these proteins require essential amino acids, some amino acids which are not synthesized in our body, but they are essential. Important component of the, vital component of the peptide chain, because they will be having some particular groups, or they will make some connections with other peptide or some other thing. That's why they are very important. So, what we do? What we do to get these essential? They are not synthesized, no? So, what we do? We take these essential amino acids from external sources. From external sources. What are these sources? What are these external sources? External proteins, particularly the animal proteins. Sir, we are vegetarian. Then from where we will get? Milk. This is the most important source. Another important source of protein, those who are vegetarian, can anybody suggest? Pulses. Pulses. Dal. Lentils. Beans. Soya bean. These are beans. So they are having proteins. But one problem is there. Their content of essential amino acids are less. So their biological value is relatively lesser than the animal protein. Can you name some essential amino acids? Glycine, methionine, valine. So sulfur containing amino acids, cysteine, methionine. There are 8 essential amino acids. So in adult, there are 8 essential amino acids. So we are dependent from external sources. Understood? The globin, the macrophage engulfed the hemoglobin, I mean red cells, it destroyed the cell and liberated hemoglobin. Now this hemoglobin will be splitted into heme and globin. Globin is a protein made of amino acids, some essential amino acids are there. So now this globin will be hydrolyzed into several amino acids, and these amino acids will be released into the blood, and they will join the amino acid pool. This cell require amino acid. Okay, you take it from the pool. Beta cells of pancreas, islets of Langerhans, they will prepare insulin. And for this insulin, we need, if I want to prepare it, I need amino acids. So you take it from the pool. So now this amino acids released from the globin will go to the pool and for use, maybe for the same globin synthesis, maybe same thing in same brick may be utilized for the bricks. Same brick may be utilized for this building, some other building, for roads, for various purposes like the amino acids, these acids may be required in different bodily processes where these proteins will be synthesized. Now what will happen to the heme? And this is happening where? Within the macrophage. Remember this. Now this is happening within the macrophage. Macrophage hydrolyze the globin, release the amino acid into blood. Now what will happen to the heme? Again, the heme will be splitted. Heme will be splitted. Can you remember the structure of heme? Yeah. What is this ring? Pyrrole. Hello, what is this ring? How many pyrrole rings are there in hemoglobin? 4. How many? 4. I will not go in detail. These are the 4 pyrrole rings, and these pyrrole rings are joined together. They are joined together by a breeze. What is this breeze? Right. Good girl. This is methane breeze. Methane breeze is like this. CH double bond. CH double bond. So in this way, battery nahi. Amar battery. It is night. Disturbance. Hello. Here. So now these pyrrole rings are attached to one another. Here is the iron. We know heme contains iron. So they are linked to each pyrrole ring by 4 valences. It may be there in exam. I don't know. And it remains in ferrous form. If it remains as ferric form, what will be the name of this compound? Methemoglobin. Then it is no more hemoglobin. It is a useless hemoglobin if the iron remains in ferric state. The name is methemoglobin. So every detail you have to know while you will be going through. So because some question may be there, like this. So you should learn T. Now it is him. Now, suppose this is the A and there are some side chains. Methyl, vinyl, propyl. This type of side chains are there. So now, within the phagocyte, what it will do? The phagocyte, what it will do? They will, another valency is combined with the globulin, and there are 2 beta chains and 2 alpha chains, and this is globin, 141 amino acid alpha, 146 amino acids in each chain, beta chain. Every chain can bind with one heme. So this is attached with this. This require one heme, this require one heme, this require one heme. So one molecule of hemoglobin will have how many heme? 4. Will have 4. So when the macrophage splits one molecule of hemoglobin, it will produce 4 chains, means globin, and 4 heme moieties. So ultimately, when it is splitted, the bonds are separated, broken, then we will get this heme part. Now, the macrophage will go for next step. What is the step? Hey, you get out of here. So iron is released. Where? From the macrophage. Go out of my cell. Now you cannot. Where it is coming now? In the blood? From here it is coming, released, macrophage released it into the blood. Now it cannot travel in the blood alone. It needs a carrier. Apoferritin. It will bind with this iron and bring it to the liver. Bring it to the liver. Liver will transform it into transferrin. Iron will be transformed into transferrin and there it will be stored. Now, the bone marrow requires iron, so it will take this iron from the store, from the almirah. So, from there it will draw, means liver will release it into the blood. From blood, the bone marrow will take this iron for heme synthesis, hemoglobin synthesis. So, it will be recycled. So, this iron will be recycled. So, after losing this iron, these 4 pyridines left, now it will split one bond. Now what will happen? It will become straight. Instead of this, it will become straight. Now we call it biliverdin. Now we call it biliverdin. Now this biliverdin will be converted into bilirubin. Will be converted into bilirubin. Which cell? Who did it? This cell. What is the name of this cell? Macrophage. So ultimately, it released amino acids and released iron and released and produced biliverdin. And biliverdin will be again changed into bilirubin. Then it will go to plasma. My function is over. So now the cell produced biliverdin. So this will be converted into bilirubin, free bilirubin, unconjugated bilirubin, and will be entering into the plasma. It is happening in spleen. So ultimately this bilirubin from spleen will be entering into plasma. Into plasma. Now we have to see what will happen to this bilirubin. It is free, no? It has to be transformed into conjugated. It has to be for elimination because this conjugated bilirubin is soluble, and so that it can be excreted. So it needs bilirubin needs conversion into conjugated one. So where it will be done? this will be done only in liver. So from all sources, the produced bilirubin will be coming to liver for further modifications into conjugated one. Okay, now this conjugated bilirubin, liver will get this and liver will make it conjugated with another glucuronic acid. When it combines with glucuronic acid in liver, in hepatocyte, it is transferred into conjugated protein, conjugated bilirubin. Now it is soluble. Liver, one of the important function of the liver is it produces bile. It produces bile. Now liver will secrete this conjugated bilirubin into bile. So from liver, bile is coming and storing in gallbladder. So when we are taking food, this has to be digested. Gallbladder constricts. Pressure within the gallbladder of bile is increased. So now bile will be entering into the duodenum through a duct. We call it common bile duct. So through this duct, bile will be entering into the duodenum and it will, bile has some functions. It provides so many pigments, it provides bile salts, everything has got a function. So ultimately, this conjugated bilirubin is entering into GI tract. Okay, now it has entered into intestine. Here, it will face, we have some beneficial bacteria within our intestine. This bacterial enzyme will convert this conjugated protein into urobilinogen. And some of these urobilinogen will be again reabsorbed, from lumen into the blood. And again it will come into liver. And liver will again send it to bile. So, liver, bile ducts, stomach, liver, gallbladder. So it has synthesized what? Conjugated bilirubin. Now this conjugated bilirubin will be liberated from the liver into the bile, and this bile will be stored here. So when we take food enters into the duodenum, it contracts by a local hormone, influenced by a local hormone, cholecystokinin. So this will be released here. So ultimately, conjugated bilirubin is coming here. So it will pass and face the bacteria in the intestinal mucosa. They will convert it into urobilinogen. Okay, and most of these most part of this, 80% of this will be passed out of my body through stool as starcobilin. As starcobilin. Okay? Now what will happen to the rest 20%? So, in the GI tract, lot of capillaries are there. Capillaries function to absorb the digested foods. So, they will absorb, the rest 20% will be reabsorbed, will be absorbed into the blood. So, this blood is again coming to liver, coming to liver. Liver will again, this rest of the 20% we released in bile. So, in this way, these are excreted. Now, some part of the, conjugated protein, this absorbed 20%, no? So, out of this 20%, 10% is recirculated and rest of the 20%, it is not coming again, not coming in the, coming out through the stool. So, what will happen to that? So, this will be circulating in the system and it will passing through the kidney and it will be excreted through the kidney as urobilinogen, and when it is liberated through urine, it is now urobilin and it is giving the color of urine. And the starcobilin present in the stool giving yellow coloration of the stool. So now can you understand why our stool color is yellow? Why our urine color is light brown? This is the reason, because of presence of. So this is how we metabolize bilirubin. Now, some bilirubin produced in the macrophage cannot be converted fully into conjugated. So, some will remain free. That's why when your liver function test will be given, you will find conjugated, your report is there, conjugated protein, this mass, unconjugated means free giving is this mass. It has got significance. What does it mean? Conjugated one, normally bilirubin content, total bilirubin, including free and conjugated, it is less than 1 milligram per 100 ml. But in case of jaundice, it may rise. It will rise. When it is about 2 milligram %, then it is noticeable. This discoloration, or this is the sclera, white portion is the sclera, no? It has got elastin. So this elastin has got high affinity for bilirubin or conjugated bilirubin. So it binds with this and it gives the color. Similarly, it happens in our skin. Okay, that's why there is discoloration. There is yellowish coloration of the sclera or yellowish coloration of the skin. Jaundice is known as icterus. Spelling? Jaundice is characterized by the yellowish pigmentation of the skin, and white means sclera. White means sclera. Sclera of the eyes due to high bilirubin level. So it becomes prominent when the bilirubin content, total bilirubin content is about 2 milligram, 2 to 3 milligram %. Then it becomes noticeable. Jaundice is not a disease. The first question was. It is the manifestation of different diseases. Say hemolytic anemia. It is a disease, hemolytic anemia. sickle cell anemia, thalassemia, vitamin B12 deficiency disease. What is the name? Pernicious anemia. So in this sort of diseases, there will be hemolysis outside the liver by the macrophages. This will be damaged and they will be taken up by the macrophages. So they will convert more hemolysis, more conversion of formation of bilirubin. So they will blood levels it will be raised, and this bilirubin will enter into the liver. If the liver is not able to convert it, then again bilirubin will further rise. But if the liver is capable of converting it, so now the bilirubin is very high. So liver has to convert so many bilirubins into conjugated one. Okay, so this conjugated under this, it cannot convert all the bilirubins. It becomes beyond its capacity. So bilirubin will be raised. Okay, so there will be also more production of conjugated one. Okay, now we know there will be more elimination of conjugated protein through stool and urine. So urine will become yellowish. In operation theatre, in ICU, near about 40% of patients in ICU, they suffer jaundice. Because a lot of drugs are given, they are destroying also. So there are different reasons. And among the newborn baby, 80% suffer jaundice. Within a week, it develops jaundice. This is called the physiological jaundice. Why? Answer is here. Its liver is not that much developed. Means the enzymes are not that much developed. But within the blood or in the fetus also, it is also producing RBC. No? So it will be having certain life. So after that, what will happen? It will be destroyed. So it will liberate bilirubin by the macrophages. But this bilirubin is not converted into conjugated one. Why?
Because of the
liver is not prepared. Liver is not prepared. The enzymes who will be converting it, they are not yet available. So there will be increased level of bilirubin. So it is fetal jaundice. And the prevalence. What is there? Males are more commonly affected by hepatocellular jaundice. My origin is liver. But the females, they suffer hemolytic jaundice. Most of the female suffers. The prevalence is less in female, less in male, more in female. But in the female, it is mostly because of hemolysis, and for the male, it is because of disturbances in the liver. And depending upon age, youngs are affected by infective hepatitis, viral hepatitis. But elderly, they suffer jaundice because of some other disease. Disease of the liver means there is abscess, there is carcinoma, there is obstruction in the bile ducts. So these will create jaundice. So these are different kind of jaundice. See the metabolism of bilirubin. Just try to recall. The spleen, reticuloendothelial cells. Beside the skin, what are other organs? Bone marrow, liver also. Okay, and lymph glands. So here I am mentioning only the spleen. So within the spleen there are a lot of reticuloendothelial system cells. One of the cell is what? Macrophage. So this is the macrophage. So within the macrophage, what is happening? Hemoglobin is broken down into globin and heme, and amino acid is released into blood. And the iron is released from here and taken up by the carrier apoprotein to the stored, where they are stored. The iron is stored in the liver. So there it is stored. So when my bone marrow will be requiring iron, so this will collect from this store. My bone marrow will be requiring iron for what? For what? Sorry? For erythropoiesis, for hemoglobin synthesis. So, from him, it will produce biliverdin, then it will convert it into bilirubin. Okay, now bilirubin will enter into the. So, while traveling through blood, actually we call it free hemoglobin, but actually not. They remains bound to albumin. Anytime albumin can release them. It's not a permanent one. So that's why we call it free. So here I will be ending.